Hi everyone, welcome to Lab 3, Blood Typing. Now that you're familiar with the immune system and the interactions that occur between antigens and antibodies, we need to talk about another important application of these principles, blood typing. Last semester you studied the histology of blood, but at this point we have yet to answer several important questions. For example, how does blood vary from one person to the next? Why are there different blood types? And finally, why is understanding and recognizing these differences so important? Before we begin answering these questions, however, let's briefly review what we know about blood. Remember from the unit on histology that blood is a special type of connective tissue, and it's composed of four main things, the first of which are red blood cells. Red blood cells are also called erythrocytes, and they are responsible for transporting respiratory gases like oxygen throughout the body. The second main component of blood are white blood cells. Also called leukocytes, white blood cells are the cells of the immune system. The third main component of blood are platelets. Also called thrombocytes, platelets are cell fragments that are necessary for blood clotting. Lastly, blood is composed of plasma. Plasma is the fluid portion of blood, and it constitutes about 55% of the total blood volume. So the question to ask at this point is, if everyone has blood composed of the same four components, how is it that you and I may have different types of blood? To answer this question, we have to discuss some of the basic concepts of genetics. Remember that all of the cells in our bodies contain genetic material or DNA, which is confined to the cell's nucleus. The DNA in those cells can be arranged into dark staining structures called chromosomes, and humans have 23 pairs of chromosomes or 46 total chromosomes in each cell. Located along each chromosome at places called loci are genes. And remember that genes are important because they control specific traits, and in this case the trait we're interested in is blood type. Let's take a closer look at genes. In the human genome there are approximately 25,000 genes, and each of those genes has alternate forms called alleles. What this means is that each gene may have a slightly different base sequence, and as a result produce a slightly different version of a specific trait. For example, there are three different alleles for blood type: the IA allele, the IB allele, and the lowercase I allele. For simplicity, each of those alleles is referred to as the A allele, the B allele, and the O allele. The figure at the right represents a pair of homologous chromosomes. Notice that three genes are labeled on this chromosome. Both copies of gene 1 are drawn with the same color. This means that the DNA base sequence in both copies of this gene is the same on both chromosomes. Genes 2 and 3, however, are drawn with different colors, indicating that these genes have slightly different base sequences. Even though they're different, they still code for the same trait. So for example, let's assume that gene 2 codes for blood type. Although gene 2 is different on each chromosome, both copies of gene 2 still codes for blood type. The reason you may have different versions of each gene is because in sexually reproducing organisms like humans, each parent contributes one allele to their offspring that may or may not be like the other parent's allele. So in other words, your DNA contains one allele from your mom and one from your dad for each of the genes in your genome, including those that determine blood type. Let's take a closer look at each of the genes in this figure. Not all of the alleles in your genome are equal. For example, dominant genes are genes that mask the expression of an alternate gene at that locus. The gene whose expression is masked or hidden is called a recessive gene. With blood types, the A allele is dominant, the B allele is also dominant, and the O allele is recessive. So again, let's assume that gene 2 is the gene that determines your blood type. Again, we have two different versions of this gene. So let's say that this copy of gene 2 is from your dad, and it's the A allele, and this copy of gene 2 from your mom is the O allele. So you have one A allele and one O allele. Okay, this combination of alleles is called your genotype and can simply be written AO. But how do you know which type of blood you have? In this particular example, your blood type is type A. Remember that the A allele is dominant and the O allele is recessive. Although the O allele is present, we can't see that it's there because the A version of that gene hides the O version. The blood type that results from a specific gene combination is called the phenotype. So to recap, in this case, the genotype, the combination of genes, is AO, and the phenotype, the blood type that results, is type A. Let's do another example. What if your mom gave you the A allele and your dad gave you the B allele for blood type? Your genotype, the combination of genes, would be AB, and the phenotype, the blood type that results, is type AB. Remember that both the A and B alleles are dominant, so in this case they're both expressed. When two dominant alleles are simultaneously expressed, we say that the two alleles are codominant. What if gene 1 is the gene that determines blood type? Both copies of this gene are identical, so let's assume that both your mom and dad gave you a copy of the O allele. This means that your genotype is type OO, and your blood type is O. The only time you see expression of the O allele is when you have two copies of it. An O allele in combination with either of the dominant alleles, A or B, will cause the O allele to be masked or hidden. Just to make sure you're comfortable with this information, complete exercise 1 at the top of page 3 of your handout. Before moving on, let's take a look at question 2 in exercise 1. Question 2 says, if you have type B blood, what is your genotype? If you have type B blood, there are two possible genotypes. You can have two copies of the dominant B allele, giving you the genotype BB, or you can have one copy of the B allele and one copy of the recessive O allele, giving you the genotype BO. Now let's complete exercise 2, determining phenotype. Question 1 says Meg and her partner Mike are expecting their first child. Mike has type AB blood and Meg has type A. What are the possible blood types of their unborn child? To answer questions like this, we have to work out Punnett squares. Hopefully you're familiar with Punnett squares from general biology. If not, they're really easy. So let's work through the first question together. To get started, you have to know the genotypes of both parents, and you'll write the genotype of one parent at the top of the Punnett square and the genotype of the other parent along the side of the square. So for example, we know that Mike has type AB blood, which means that his genotype can only be one thing. It must be AB. So we'll write his genotype along the side of our square. Meg has type A blood, which means that her genotype can be one of two things. She can have genotype AA or AO. Both combination of alleles will produce the same phenotype, which in this case is type A blood. So along the top of one square, we'll write AA, and along the top of our second square, we'll write AO. From this point, we have to fill in each of the blanks in the square, and we do that by cross-multiplying across the square. So for our first blank, we have A times A, which gives us the genotype AA. In the second square, we also have AA. In the third square, we have A times B, giving us genotype AB. And in our final square, we also have A times B, giving us AB. So assuming that Meg's genotype is AA, their unborn child could have two possible blood types, A or AB. Now let's look at our second square. Again, cross multiplying, we first have A times A giving us AA, A times O giving us AO, A times B giving us AB, and finally B times O giving us BO. So in this case, there are three possible blood types, A, B, and AB. Using this information, you should now complete exercise 2 by answering questions 2 and 3 in your handout. Afterwards, follow the instructions in your handout for completing this week's lab on blood typing.